morning, everyone. Welcome to the live webinar series, Breakfast at UMHealth. I'm Peng and I'm the moderator for this session. It is my honor to introduce our speaker for today's breakfast talk. He is a pharmacist by training and an expert in molecular modeling and drug design, holding a PhD in the same field. Currently serving as the head of the Pharmaceutical Chemistry Department, Faculty of Pharmacy. His research focuses on computer-aided drug discovery. Today, he will share his insight on an exciting topic, the self-evolving peptide algorithm. This innovative method can potentially transform how we approach drug discovery. Please join me in giving a warm virtual welcome to Dr. Choon. Dr. He, the virtual stage is yours.
Okay, thank you very much, Peng. So let me share my slides. So okay, good morning, everyone. So welcome to the Breakfast at UMHealth. So I'm Choon. So today I'm going to present you a project funded by FRGS, which is the Self-Evolving Peptide Algorithm, or in short, I call it SEPA. So I'm moving to my second slide. Can you see it?
Yes.
Okay, thank you. Yeah, thank you for the confirmation. So why peptides? Yeah. So just a brief introduction. So peptides has been gaining interest, increasing interest and attentions as potential drug candidates due to their high specificity towards the target and high potency, as well as low toxicity. And they are also able to mimic the natural biological molecules. So therapeutic agents that are based on peptides are short chains of amino acids. Normally it's like 10 to 100 amino acids, or they are mimics, which is the peptidomimetics. So here I want to give you some examples of the roles of peptides in drug discovery. So I believe you all are well aware of ACE, the angiotensin converting enzyme, ACE2, which is quite famous during COVID time because it is the target entry protein for COVID to enter our body. However, there is another very similar protein, which is the ACE1, or I just call it ACE. Yeah, so it can actually regulate the human blood pressure. So, for example, during the 1970s, yeah, scientists actually discovered that the extract of a protein, which is called a peptide, yeah, can actually it is the extract from a venom of a viper snake, yeah, for information. So can actually causes hypertension. Yeah. So from here, so further research actually found out that the C-terminal, yeah, the C-terminal near the proline, yeah, of this peptide is the one that able to bind to the active sites of angiotensin converting enzyme. Yeah. So, but however, if you're working with peptide, you know that peptide have a lot of peptide bond that can be easily hydrolyzed. So it is not so stable if you want to consume it orally. So further study actually have some improvement of this peptide to become a non-peptide that eliminate the peptide bond so that it cannot be hydrolyzed by the enzyme in our body. So the first non-peptide mimics of this compound is the succinylproline. Then further leading to our first ACE inhibitor, yeah, captopril, that is already marketed as the antihypertensive drug. So, yep. The second one is the endogenous opiate peptides. So we have a few inside our body. So the common ones are the alpha endorphins, beta endorphins, gamma endorphins, as well as a few other examples that I will show you later. But try to focus on the one that I highlighted in red. So these 4 amino acids is the one that are playing a very big role in the binding of the endogenous opiate peptides towards our opiate receptors. So in this slide also, I showed two other endogenous opiate peptides, which is the enkephalin. Yeah, we have the methionine and the leucine. Yep. So you can see the same 4 amino acids is also repeatedly occurred in this smaller peptide, yeah, as our endogenous opiate peptide. So the further study actually, this is actually discovered only after the use of morphine. Yeah. So if we try to compare the structures of morphine and our endogenous opiate peptide, so the structure that highlighted in blue for both the opiate peptides and the morphine is the one that actually binds towards the opiate receptor. So if you can see, morphine is not a peptide. That's why it is so stable and can be a very potent and the painkiller that binds so strong in our body and then causes not just painkiller effects, also causes others a lot of side effects, such as hallucinations and addictions. Okay, so however, so the vast diversity of peptide sequence may hinder the effectiveness of screening for potential peptide therapeutics, as if we're searching for a needle in a haystack. So the number of peptides to be screened could increase exponentially. So 20 is the indicating the 20 basic amino acids. So it only the natural form, which is the L amino acids, which doesn't include D some more. If you want to include, it will become 40. So N is actually the number of amino acids in a peptide that we want to screen. Then here comes the game changer. So with the introductions of computer-aided drug discovery, we also call rational drug discovery. So the process of screening can actually be reduced in terms of both the cost as well as the time. So based on the computing power that you have. So basically for computer-aided drug discovery, we have two approaches. One is the structure-based, that the drug target structure, normally is the crystal structure, need to be available for us to identify the potential peptides or any small compound to be able to go into the active sites of the enzyme. The other approach is the ligand-based drug design, where normally the drug target is maybe unknown, yeah, or the crystal structure is not available. And we'll be using a group of similar structured compounds that giving the similar biological activity. And from there, we come up with some algorithm, which is called a quantitative structure activity relationship. Then from there, we got information to map with those small compounds that we want to screen. So these two approaches can be applied for virtual screening. And at the end, we can select potential compounds based on either their binding affinity towards the drug target, if it is available. If not, maybe how similar it is compared to the known inhibitors or ligands, and as well as like the availability of it in the market, or how the compounds actually behave, whether it is behave more likely as a drug. We call it the drug likeness properties. Yeah. And it is a very good approach to identify lead compound. We call it. It's not the final compound, of course, the lead compound, which can cause some biological activity. From the lead compound, then we have we need more time for it to be optimized, yeah, into a real drug to be marketed. So what is SEPA? So it is a self-evolving peptide algorithm comprised of repetitions of automated peptides generations or elongations, and followed by molecular docking, which is a structure-based drug design approach. and a thorough interaction analysis of the generated peptides towards the drug target. So these icons are the software, the important software we are using in SERPA. Okay, so how does it work? So the first one is a PyMod. It's a software to generate peptides from the 20 basic amino acids. So this is the first version of SERPA. So we are just using 20. So maybe in the future versions, we can use more type of different amino acids. Then we have the OpenBabel, which is a structural optimizations of the generic peptides, because when the peptides is generated by PyMod, it is actually linear. So we need to try to optimize the structure so that it's resembled as best as the nature form. Then we have these Autodoc tools, which come under the package software MGL tools. So it is used to prepare this peptide to be dockable peptide, as well as a drug target, so that the next software, the Autodoc, we now recognize both your input files, which is the small molecule, which is your peptides, and the macromolecule, which is your drug targets. So VINA is to perform the docking of the small molecule towards the drug target. And then we have HP Plus, which is a hydrogen bond analysis software that we use to program our own algorithm of interaction analysis. And all this is being run in the Ubuntu operating system, which is a Linux operating system. So from there, we actually programmed the bash shell script under Ubuntu and to communicate all this software together, yeah, so that at the end they can rank the peptides based on the interaction analysis and select the highly ranked peptides or subsequent elongations. So this is a graphical abstract for the algorithm. So let's say we started with dipeptides, because actually amino acid, single amino acid itself is not so worth it to start with because it's too small. So normally we start with dipeptides, but if you want to run single amino acids, you can also start with that. So, but I've showed the example that we normally start with dipeptides. We have 20 times 20 basic amino acids, so you have 400 dipeptides. And from here, you will perform a docking towards the drug target you want. And then the top 100 candidates will be selected based on the thyroid binding interactions, as well as their binding affinity towards the drug target. So this top 100 candidates will be further elongated with the basic 20 amino acids residue, and then we'll produce a 2,000 new elongated peptides, which is a tripeptides. So these 2,000 tripeptides will further be subjected to molecular docking again to the same drug target. And then the top 100 candidates of the tripeptides will be further elongated too. So this project will repeat until when the length of the peptides that you defined in the software, you can actually define what is the amount of amino acid that you want to produce for the peptides. Yeah. So this is based on the natural selection, so the survival of fittest concept. So the fittest individual in a particular species, yeah, will able to survive this algorithm, and of course the weak ones will be eliminated. Yeah. Okay, so it's free, yeah, for now, the algorithm. So you can actually scan this QR code, and then the instructions, the script, and prerequisite files are all there. Okay, so we have managed to publish one paper for this algorithm. Okay, as a summary, so the optimizations of SEPA is still needed by testing with various reported peptide inhibitors, since because our group also have limited human power. Okay, and then SEPA provides an alternative automated approach for the virtual screening of peptide sequence against drug targets. Result with higher accuracy could be achieved by employing the important interaction between ligand and drug target observed in the reference structure for Tara interactions analysis within SEPA. So this Tara interaction analysis is the one that major thing that we programmed ourselves with a lot of optimizations. So the important thing is that we need to really identify the important, or we call the essential amino acids that give, let's say, the strong interaction, such as hydrogen bond interactions or ionic interactions at the active sites, when you try to understand how the natural substrate or the known inhibitors is actually binds at the binding site. Yeah. And future implementations of AI as part of the docking analysis process could further increase the accuracy and reliability of SEPA. Yeah. So one important note that we need to take into consideration if we want to work with peptide is that they are easily degraded. That's why it cannot be taken orally as drug directly, and difficult to cross the intestinal mucosa. And for peptide drugs, it may also induce immunogenicity when the number of amino acids increases. Thank you. That's all from me.
Thank you, Dr. He. Thank you for the insightful presentation. And we will now open the floor for Q&A. There's already one question. Maybe I can read it for you.
Okay.
Does this mean that a host of new drugs could be developed based on this system?
Host.
I think he meant the target, maybe.
I don't quite understand what does it mean. The drug target of new drugs can be developed if
I think that is what he or she means.
That is maybe a reverse type of algorithm. So it means that you are going to use a drug with to identify the unknown drug target using this algorithm. For now, because we are just focusing on generating small peptides, and normally the drug target is very big, it's a few thousand amino acids. So I think for now, this algorithm a lot. Okay. Okay. Yes. A lot. Yeah. Yes. Definitely. A lot of peptide space, maybe drugs could be developed. I can say that if we can improve the algorithm by implementing AI and have a better interaction analysis algorithm, definitely we can find a lot more useful, maybe useful peptides is good enough, which is because they are not so expensive to be produced, and then they are also more selective, more potent, and less toxic. Just that if the drug delivery person can overcome how to deliver the peptide through orally, it will solve a lot of problem. Yeah.
Okay. While waiting for more questions, maybe I can also ask you a question.
Okay.
Does this algorithm has a GUI, graphical user interface?
At this moment, not yet. Yeah. You will need to follow the instructions in the Google Drive that I shared just now. So the instruction is all there, and it's just simple. How to say, if you have some knowledge in Linux operating system terminal, it's not very hard for you to run it. Yeah, just maybe the troubleshooting when you have errors come out. So by right, if you just follow instructions, you're able to run something. Yeah, the problem is that the preparations of the drug targets might be challenging if you're not familiar with it, and maybe the preparations of peptides. So, but if you are a orthodox Vina user, so I believe you have no problem to do it. Yeah, but that's a good suggestion. Maybe in the future we can include a graphical user interface. Yeah.
Okay, we have 1 minute to 9. If you have any more questions, you can still type in the chat box or Q&A. I think there's no question.
Okay.
Okay.
So thank you.
Since there's no question, we will end this session now. Okay, once again, thank you, Dr. He, for your presentation and everyone for joining us today.
Yeah,
We hope you found this breakfast talk insightful and inspiring. Have a wonderful day ahead. Thank you.